So today I'd just like to start off with the topics we're going to cover. So we'll start with tips for optimizing your nuclei extraction protocol. So we'll start with what kind of tissue you're working with, how to get that initially set up, and then how to actually go through the extraction so that you can optimize the yields and the efficiency of your extraction as you do it. Once we've completed the extraction part, we will actually move to a real-time demo. So I actually have a nuclei sample here that I will load onto the Celldrop automated series of cell counters and count here for you and show you all the tips and tricks to analyze the sample on the Celldrop with all the analysis tools that are built into the software. And then after that, we will move on to a Q&A session with myself and some of the other application scientists that are on the panel today. Before we get there, let's start with a poll question. So I would like to understand what types of tissues you analyze. So that could be fresh tissue taken directly out of, an animal. There's frozen, frozen could be, one of those preserved tissues, like a fresh that you preserve through freezing. There's FFPE preserved tissues. Of those options, do you use human tissue? Do you use non-human mammal tissue? Do you use plant tissue? I think we've conceived of everything there for possibilities in terms of nuclei extraction. So I'd just like to get a feel for what our audience today generally works with. Looks like a few more results kind of creeping in here. So I'll give everyone a few more seconds to finish answering and close the poll up now. So it looks like we have a heavy bias towards fresh tissue, but we have a good representation of frozen and FFP preserved as well. And it looks like we have a lot of representation on human and non-human mammal, as well as some plant people. So it looks like we got a little bit of everybody today. So hopefully I can answer everyone's questions, but if not, we have an opportunity to get to that later. So as just an overview of what we're going to do today, our webinar map, if you will. So we'll start with tissue prep. So preparing your tissue, whether it's fresh or FFP preserved or frozen, getting that ready for the actual extraction process. Then we'll move to the overview of the extraction, that sample for quality control analysis on the Celldrop. After that, I will show you the counting protocol, so all the different settings that the Celldrop can use to best quantify the nuclei to your expectations. We'll talk a bit about imaging and nuclei counting and the different ways to do that and what the Celldrop can offer there. Then for the analysis portion, I will actually do the live demo. I'll show you how to count a sample and analyze it with the powerful, intuitive tools in the Celldrop software. And lastly, we'll cover downstream applications. So now that you have your nuclei, what are you going to do with them? I'd like to point out that throughout the webinar, if you have any questions, please submit them to our panel. We have some application scientists on standby on the panel to answer them as they're submitted, but I also will plan to answer a number of questions at the end of the session as well. So we'll see. But with that, let's dive right in. Let's start our journey at our tissue preparation step. So as we've kind of talked about a little bit already and alluded to with our poll question, you want to prepare your tissue, whether it's fresh, frozen, or FFPE fixed, to analyze it appropriately. So, we have a lot of options that we've worked with. We've worked with human, rat, mouse. We've had customers that have worked with tomato root and corn and other samples, and just a whole variety of different sample types that can be utilized. Some considerations to take here are if you have a really big tissue, even if you're extracting or grinding it up all in one spot, it might be beneficial to section that large tissue into several smaller pieces to go into the same tube to help it more efficiently extract. But your protocol will cover that in detail. So now that we've prepared our tissue, we will move on to the extraction phase. So this is where we're actually going to isolate our nuclei from the tissue. We have personally worked with the Miltenyi extraction buffer and GentleMax system. So the GentleMax has been great for us because it automates the actual grinding process. So something that I would have done manually years ago in the lab, this equipment does for you, and they provide all the buffers and filters. So that's been a great way to process a lot of samples simultaneously. The 10x Genomics kit is another kit that we have used. It's a more manual kit that involves the manual grinding, but it's still an excellent extraction system. We have some images later in the talk that I can show that demonstrate how efficient that is on the Celldrop. And lastly, we have our user formulated buffer option. So there's, that's something that we generated in the lab here, just looking at stuff that was in the literature. And that also has worked really well. So the nuclei can be extracted in a number of ways. This probably doesn't even cover all the different options, but these are 3 that we've tried personally, and they've all worked really well on the Celldrop. So I have a montage that'll play as I kind of describe what's going on here for you of the nuclei extraction process. So you can see here that we're using the Miltenyi GentleMax system. And right here in the video, I just put a cooling sleeve on top of one of my samples. You can see the system is doing the grinding for me, which really helps to make sure that all my samples are processed the same way and one isn't processed differently than another. But the importance of that cooling sleeve that I put on in the video is one of the most important considerations in the nuclei workflow is keeping everything cold. So the nuclei are protected generally by the cell membrane. And when we isolate them, we release them from that cell. And what happens is they're a little more fragile. So we want to move quickly in this process, and we want to keep everything cold to slow or prevent osmotic lysis of the nuclei. We want as many of these to make it through to the sequencing step as possible. The best way to do that is through cold and also working quickly. To help you work quickly, I advise that you prep everything well in advance. All your dilutions should be done way ahead of time, and you want to make sure all your buffers are chilled or warmed to the appropriate temperatures according to the instructions for the kit, to be sure that you don't have any wait time in your protocol, because that will affect your extraction efficiency if you're pausing to dilute something. And then lastly, a couple of the steps in this video montage that you've seen, I've been pouring samples through filters. That's really important to understand what is going on with your tissue and what filtration steps are going to be needed to remove as much cellular debris as possible. We don't want a lot of debris when we get to the sequencing phase, so removing that debris beforehand is important. And different tissues might require a different pore size on the filter. So that's just, those things you want to consider here. So with our extraction, our tips here on the extraction completed, we will move on to talking about prepping our isolated nuclei sample. So we now have our suspension of nuclei. We want to prep it to count for quality control analysis on the Celldrop. So the first step here is equilibrating your AOPI to room temperature. Definitely keep the nuclei suspension on ice. We want those to remain cold, as I noted before, to ensure that they are stable. But the AOPI will perform better when it's warm to room temperature. Like all fluorophores, the cold can kind of quench some of the signal, so we want to make sure that there's no signal loss from just being cold. Once the AOPI has sat out at room temperature for a few minutes, we will add that to label our nuclei in suspension, so a 1-to-1 mix, similar to trypan blue method. So you'll just take, in a fresh tube, you'll put a small volume of your nuclei isolation and then an equal small volume of the AOPI, and pipette up and down a few times to mix. Before you move to the counting procedure on the Celldrop, if you maybe set the tube down to do something and come back once it's been in the AOPI, it's really important that they are thoroughly mixed so that everything is homogeneously resuspended before it's loaded onto the Celldrop. I would like to point out that there is no incubation time required for AOPI, so it's not something you have to rock for 5 minutes or anything like that. It's simply mix 1-to-1 and input on the Celldrop. There's no wait time needed. I would like to point out, I have been talking a lot about AOPI, but trypan blue is something that can be used to count and quantify your nuclei. But the combination of AOPI and the fluorescent signal of the two stains removes a lot of subjectivity from our counting and offers a superior accuracy in the count. So trypan blue does work. We just strongly recommend the AOPI considering the nature of the sample here. So again, I'll show you another brief montage of prepping our sample here. As I've pointed out, the first tip is making sure that your AOPI is appropriately warm to room temperature. It doesn't take too long, just a few minutes, but it is important to do. It's ideal to minimize your volume of nuclei counted. A lot of times these don't yield a lot of sample volume. So you don't want to take 10% of your 20% of your sample just so you can do a nuclei quantification. So we actually have options on the Celldrop where you can use less than 10 microliters of your nuclei sample to make sure you maximize how much you're inputting on your final analysis. When you're resuspending the nuclei, you want to be thorough, but you also want to be delicate. The nuclei, as I mentioned previously, are very fragile. So if you vortex them too heavily, you can pop them. I typically recommend just pipetting up and down a few times or very gently vortexing. And then once everything's mixed with the AOPI, I recommend moving quickly so that you can get the sample processed quickly and move on to all your other various samples and then your sequencing as fast as possible to minimize osmotic lysis. So now, before we get into the actual quantification, I'd like to give you a little bit of information about the star of the show today, the Celldrop, that allows us to count these nuclei or cells without slides. So the Celldrop is a precise and reliable nuclei quantification method, and it's critical for QC in this extraction workflow. It's an automated counter, so it's an image-based automated counter, and it has no consumable required. And it can take advantage of these fluorescent AOPI labels. It's to provide fast, reliable, and precise nuclei counts every single time. There's a few key features of the Celldrop I'd like to cover. Most importantly is that direct pipette. So direct pipette, as I mentioned, requires no plastic slides, so there's no plastic consumable. That's great for any green initiatives at your institution. And it's also great for your budget, and you don't have to worry about remembering to have another piece of plastic in stock to do your actual experiment. But the direct pipette offers a performance benefit as well. Because the Celldrop is using very flat precision sapphire surfaces and a high precision motor screw to control the arm, the height, there's less variability in Celldrop from measurement to measurement than you see with a traditional plastic slide or a hemocytometer. So yes, it's green and that's great, but it also offers superior performance. So it's a win-win for you in that respect. The Celldrop, using those sapphire surfaces I mentioned, has incredibly clear optics. So you can see an example image off to the side there that highlights our dual fluorescent and bright field imaging all together. So that dual fluorescence, I'll cover a little more in a minute, but it offers very powerful analysis in a very fast manner. And the Celldrop also has a ton of modern conveniences you'd expect on any modern lab hardware. So you have your exporting and your touch screen here to integrate into your lab. So a little more about direct pipette. You can see the graphic off to my side here is showing the sample. It's very similar to a hemocytometer, right? I sometimes say it's a hemocytometer with a touch screen. So if you've ever loaded sample on one of those before, it's the same process. You simply pipette 10 microliters of sample between the two surfaces. So you'll load your sample, as I just showed you. Then you'll press the count button to analyze your sample. And once the count has completed, you wipe clean with a simple Kim wipe from your lab. And that's it. That's the whole analysis procedure on the Celldrop. So in general, this takes less than a minute and allows you to quickly and precisely quantify a large number of samples so that you can move on to your downstream applications with confidence that you have quality samples to put into the sequencer. So with our samples prepped and ready to go for quality control, I would like to cover a few more features of the Celldrop. So I mentioned that there's a lot of features in our software. The counting protocol is a big one. So the Celldrop offers a variety of parameters that can be tuned from sample to sample. I would like to point out that for the most part, our default protocol works very well with most samples we've worked with, but it's nice to be able to customize any protocol to fit your exact needs. So we know that we can't possibly test every sample type, and we give you the tools to customize it for your needs. So you see here, there's a list of parameters that you can change to make sure it counts according to your expectations. The other criteria of note in the chamber height heading on that protocol screen, you see that we're using a 100 micron, that's standard height it's marked. That's what you're used to if you've ever used a hemocytometer before. That's a 100 micron height. That's a 10 microliter total analysis volume. But the Celldrop offers a 50 micron volume. Because we're not using a fixed plastic, we can adjust the volume using that high precision motor screw I discussed earlier. So as long as your sample isn't super sparse, you can take advantage of this 50 micron chamber height. where you can load as little as 2.5 microliters of nuclei combined with 2.5 microliters of AOPI. Now we know in practice it should be slightly more to build in some cushion for our pipetting, but you can use a very, very small volume of your overall nuclei isolation and save as much for your downstream applications as possible. Once you've customized the protocol, these are easy to set, save, and reload amongst the lab, so it increases precision across the lab by allowing everyone to use the same count parameters. And speaking of all these count parameters, we do have a page on our website, which I'm highlighting here, that has a variety of different protocols that we've worked with scientists in our community, as well as maybe developed in our lab to provide different starting points for different tissues. So if you work with lung, even if it's not rat lung, this protocol that is on the screen here might be a great starting point. Similarly, the tomato root protocol might be a great starting point if you work with other kinds of plants. So we have protocols on here. This list is not intended to be exhaustive. We always want to collaborate with more people and add more protocols to this page to give even more people tools to analyze their software. But this also brings me to another key point of Denovix, of what we offer for you, is the application science team is here and ready to work with you and happy to help generate these protocols and tune samples with you as needed, and provide advice and guidance to make sure that your quality control analysis for your nuclei isn't what's holding you back. So we're very responsive and glad to work with anybody that has questions. So now that we've kind of discussed the protocol and you have a bit of an understanding about that, let's talk about the actual imaging. So what the actual analysis process here. So manual nuclei counting with a traditional hemocytometer would look something like this. This was a sample that we imaged here in our lab. This is I think a mouse kidney sample. And you see that there's a lot of dark objects on the screen, and that's the trypan blue stained nuclei. So traditionally, trypan blue would stain dead cells because they have a compromised cell membrane that allows the large dye to pass into the cell. We have removed the cell membrane in the nuclei isolation, so all the nuclei should stain dark. This has been a great analysis tool for a long time, and it's been used for lots of clean tissue culture samples, and it does work really well. The dye is excluded by live cells and goes into dead cells, as I mentioned. But some problems that we know about with trypan blue are that it crystallizes over time. So you can see a few of those crystals in the image on the screen here. But the downside is if you have a really precious volume of nuclei and you load a bunch of it onto your hemocytometer and maybe your trypan blue isn't super clean, well, it's too late. You've now wasted a bunch of nuclei and there's a bunch of debris that's confounding your analysis. The other problem that we've run into with trypan blue when counting cells is that it's unable to discern live cells from RBCs. So as I said, live cells with an intact membrane exclude the dye. Red blood cells would have an intact membrane, but we usually don't want to count them. Now, I don't know how many will make it through a nuclei isolation protocol, but they could be there, and you don't want them to be, affecting your extraction efficiency data in an unintended manner. The other thing to point out is that any debris from your extraction, so some of these tissues will have a lot of debris from the actual grinding process, even after filtration. A lot of that debris will appear dark and look like a dead cell. Even to some trained users, it's hard to discern, is that dark object that's circular a dead cell, or is that some debris left behind from my extraction? So those are all challenges presented by counting with trypan blue. And that is one of the reasons that we push the AOPI for counting nuclei. So a background on AOPI, and you can see the nice cartoon on the screen highlighting what I'm discussing here. AO, acridine orange, and PI, propydium iodide, are both fluorescent nucleic acid dyes. So AO is actively transported into live cells and fluoresces green. And PI behaves similarly to trypan blue in that live cells exclude the dye due to its large size, but cells with a compromised cell membrane undergoing apoptosis will include the dye into the nucleus and fluoresce red. So both stains require nucleic acid to bind with to fluoresce either green for AO or red for PI. So that's for cells, but if you flip it for nuclei, what we're talking about is, like I mentioned before, we've removed the cell membrane. So anything that's been in any nucleus that's been extracted from a cell should light up red from the PI. And any cell that was not properly lysed in our extraction will stain green. Now, you see on the right that we have AO and PI in any dead cell, which makes sense, but we don't have to worry about compensating or any other complex balancing for the green or AO signal, because there's a fret interaction between AO and PI where the green from AO is quenched through fret by PI. So we have a binary choice of green or red. Isolated nuclei is or red, intact cells are green. There's no in between, there's no crosstalk. So instead of viability, we're going to give you an extraction efficiency number using those parameters. And notably, I'd like to point out that I've emphasized nucleus and binding DNA. What that means is any object that does not have a nucleus, so a red blood cell or any debris left behind from your extraction, as you see here, there's a lot of debris in that example image. None of it is fluorescent because it does not have a nucleus because it's not a cell. So we ignore it automatically just using these two stains. So it's a really powerful analysis tool that takes a lot of subjectivity out of the cell counting process. So I'd like to pause for a poll question before we go any further. And I'd like to get a feel for people who are attending. What are your downstream applications? What are you going to do with these nuclei once you've isolated them? Are you going to do ATAC-seq, RNA-seq, flow cytometry? Is there something we haven't considered? If you wouldn't mind entering that into the panel questions, it'd be nice to know what other techniques are out there that people are using for these isolated nuclei. I do see some results still coming in, so I'll give everyone a few more seconds here to answer. But looks right now like we have a landslide winner. I'll reveal in a minute, but I think everything's kind of halted here. So let's end the poll. And the majority of you seem to be doing RNA sequencing, but we do have a good number of people that are doing flow cytometry or ATAC-seq with their isolated nuclei. So the preference seems to be RNA, but we have some other representation here as well, and a little bit of other. So again, I'd like to know what that other really is. So if you wouldn't mind telling us, it would be great. So now we've kind of talked about the imaging and the counting. Let's actually move on to a demo to highlight how the Celldrop can most effectively count and quantify these nuclei. So I will step over to my demo instrument, my Celldrop here in the lab. And I'm going to work on resuspending my nuclei. So you can see here on the screen that we have a number of applications that the Celldrop can use to count cells. We can use the Celldrop for AOPI quantifications of tissue culture or cell isolations. We have tryptophan blue counting methods. We have even GFP methods, but today we're going to focus on this nuclei AOPI application. So you simply tap it to load it, and this is our live preview screen. So you also, now I need to load my nuclei. So while I was talking, I was resuspending my sample. I have 10 microliters in my pipette. And I'm going to, just like the cartoon showed a minute ago, put the pipette tip in the groove and pipette it onto the Celldrop. So these nuclei were isolated today. And you can tell we had a really good isolation. We have a lot of cells here, or a lot of nuclei, excuse me, on the screen here. And we can pinch to zoom, we can look around, we can just check on the sample as it's moving. So we can see that it's still settling, and we want to give it a few seconds to do that since we are going to take multiple images and overlay them. In the meantime, it's a good idea to name your sample. Everything on the Celldrop is automatically saved, so naming it will help you keep track of it later. And the first thing I would recommend doing is changing over to our red fluorescent channel, our PI, or what we expect to see, or where we expect to see our nuclei. Since this is where a majority of our sample will be, we are going to use this channel for focus and exposure. Now, I used the Celldrop earlier today to quantify this sample, make sure we had enough for the webinar, and you can see that it is still in the correct focus position, and it's still bright enough. I wouldn't change the exposure or the focus here. So you see that it's not necessary to do that every single time, unlike something that requires a plastic consumable or even a hemocytometer. There is variability between every single slide. In this case, we're not using those slides, so we don't have that variability problem. Once it's focused and exposed, it generally needs very little tuning. But if you wanted to make those adjustments, our focus button here, we have autofocus. We also have manual focus. So if you need to just tune it a little, and we also provide guides. So if you're not sure how to focus, we should give you some hints. Similarly, we want to make sure that everything's bright enough. If you've ever done any kind of fluorescent imaging, you know that you want to minimize background signal. So you want to have as little noise in the background as possible, but you want to have as much signal on the sample as possible without oversaturating. Again, I think our exposure is fine. I'm not going to tune that at all, but we provide guides again if you're not sure of what to do. Considering all that, we're going to move over and check our green exposure. I don't expect to see a lot here because we're expecting our isolations to go well. I see maybe one object. That's good. I don't want to make any more adjustments here because it'll affect every other channel. So I'll just return to my bright field image, check that nothing's moving, which it's not. I will use the default protocol, which, as I mentioned, is good for most samples, and I will press count. So our count process should take around 10 seconds. It takes 3 images, a bright field, a green, and a red image. It overlays them and counts them. So in this case, our red image will be used for approximating our nuclei or counting our nuclei. Our green image will be used for counting any cells that didn't make it or didn't quite get lysed in our extraction. And our bright field image isn't counted. It's just there for reference so we can see the morphology of the objects we're counting. Now you can see, again, I mentioned earlier, we had a really good extraction. So we are seeing 28 million nuclei, which is great. That would require probably some heavy dilutions before we go off to sequencing, but it's a good problem to have today. We have 100% extraction efficiency, which is also, fantastic to see. And we have 5,000 events counted. So you can see here the nuclei plus cells. We have 5,000 total events on the screen, which is statistically very representative of what would be in the tube. Using the cells per mil and the nuclei per mil numbers, we can calculate an extraction efficiency of 100%, like I said. And right here at the bottom, the number everyone probably cares most about is our 28 million nuclei per mil. Now, you can zoom in on this image to just see how it was counted and make sure that you agree with everything. But it does generally look very good. You can gate by size. So you can click that little histogram. You can show the distribution of cells per micron diameter. And if you wanted to truncate that further to exclude anything that you don't want in your analysis or extend it, depending on your application, you can do that. And let's say you wanted to maybe tune this protocol. So let's say you were zooming in and looking around, and there was a substantial population of cells or nuclei that you wouldn't have counted the way they were counted. We offer this optimize settings button, which this should look familiar from a previous slide. You can go through and make some adjustments here and then apply that new math to the images on the previous screen. We're not going to take a new image. We'll just reprocess with different math. And we'll do our best to work with you here to tune the protocol to best count the cells or the sample the way you would count the sample. Once you've made those adjustments, you'll be presented with an option to save that protocol. So once you've done the hard work of tuning it, you can save it and load it later, or anyone else in your lab can use the protocol that you have tuned to count your sample later or count the same sample another day. So you always have access to the same exact settings and you can use them over and over again, which is great for standardizing protocols across the lab over the duration of an experiment. Another important consideration in nuclei isolations is how many clusters or clumps of nuclei you have. So the Celldrop can also help you visualize that. Right now, you see we're in the single sample view. If you change to the graphs view, you are presented with, here's the extraction efficiency graph, but we want to change it to cluster size. So this will show us how many clusters and how many cells are in that cluster on the screen. So you can see here that we have 2,553 clusters. Really wouldn't be the right word because it's one cell. So it's of individual cells, we have 2,553 of those. And then we have 636 double cells. So it's 2 cells that are stuck together, but we can tell that they're 2 cells. So we've counted them as individual cells, but we're telling you they're stuck together and touching, which this can cause some downstream issues during your library prep. So this is a great graph that enables you to make decisions about the quality of your sample before you move on to the actual library prep phase. If you had a really high number of clusters and a really low number of single cells, you may want to look into other options to better homogenize that sample and reduce the number of clusters in there. But you do get that tool right here to analyze it quickly on the Celldrop. And then lastly, you might want to know how you can access this data later on your computer, or show your PI or show some colleagues. You can click this export button here and you're presented with a list of options. You can get a results CSV. So this will just be something you can open in an Excel file. You get a PDF report where you can include images and histograms like I just showed you. And you also get access to the raw images. So these are just exported as .png files. They're easy to access on any computer. And you can do any other downstream analysis that you may want to do using those images. So that's all, those are all options you have here on the Celldrop. And when you're finally done analyzing your sample, you would just hit next count. So we'll return to our live preview screen, and I'll show you how to quickly clean the sample surfaces. So it's quite an easy protocol. So you simply lift the arm, and then using a dry, clean Kim wipe, you apply firm pressure to both surfaces. And when I lower the arm again, you can see that the live preview is quite clean. Any small amounts of debris like that can be flushed out with ethanol, but also are generally flushed out by the next sample. So in this scenario, all of this would be flushed away by the next sample. So that, we're ready to go for our next sample. And if I wasn't doing a demo, we probably could have processed 10 samples by now. So you can see that the Celldrop counting process is very efficient and very quick, and allows you to get a lot of powerful analysis in a fast manner to move on with your actual downstream application. So with our live demo complete, and with our nuclei isolated, quantified, and analyzed for any other considerations before you move on to your library prep, we want to highlight a couple of other options here. So of course, we had a nice sample to cooperate for the webinar. We want to highlight some images that we have generated, or some of our customers have generated, that show all the different facets of nuclei isolation protocols. So we'll start with this first image here. This is fresh kidney, so not frozen, not FFPE preserved. You can see that there is a good amount of debris in the sample, but not too much. That's another thing that the Celldrop allows you to do in real time when you're looking through the image, or when you're looking at these images. You can analyze visually for debris and say, there's too much in there, I need to do further filtration or purification before I can move on with library prep. Or in this case, maybe you're happy that level of debris isn't too bad. You can see that we have a nice distribution of single nuclei and very few intact cells. And that was using this 10x genomics kit. Similarly, we did a frozen kidney, and this was using the Miltenyi GentleMax system and their extraction buffers. You see a very similar level of debris. Again, I don't think this is too much debris to move on. And this level of, the nuclei are mostly single. There's very few nuclei that are stuck together, and very few intact cells. So this was another extraction that proceeded very well. And you can quickly, within a minute, make all these decisions about this sample and move on to your downstream application. This was a frozen lung sample that we processed here using our homebrew buffers, similar to the buffers I used for today's extraction. Again, these were just generated using some protocols from the literature. The next sample is from one of our botanists, and that is a fresh tomato root sample. This was one of their homebrew kits. This is something that they came up with to extract nuclei. Again, you can see that there is some debris, but not a ton, using the images, very few intact cells. And even though there are a variety of shapes of nuclei on this sample, they're not all perfectly round. We do a really good job of recognizing the individual nuclei or the clustered nuclei and counting them appropriately for you. And finally, we'll show a frozen mouse brain sample, which brain samples in general have just been the most debris-laden samples that we have. You can see a large amount of background debris in the sample. And you do see a good number of isolated individual nuclei. I don't see any clumps of nuclei, but the level of debris is such that I probably wouldn't want to move on with my library prep until I've done further purification, because the that's going to get in the way on the microfluidic. You may even clog your microfluidic by loading this much debris into it. So before we move on, we can use a purification kit to just purify out the nuclei. So Miltenyi offers a anti-nucleus microbead purification kit that takes advantage of magnetic microbeads that are quite small and allows you to essentially wash away all your intact cells and debris. So this same sample, this is the same sample. It might be hard to believe it, but this image that just kind of moved onto the screen is the exact same sample after we ran it through the purification kit. So you can see that it's highly effective in removing debris from the isolation, as well as any maybe intact cells that were left behind or other clumps. And it doesn't affect our yield. There's still plenty of nuclei on the screen. There's still a good concentration. It's just our extraction efficiency has been improved by purifying with this kit. If you want more information about the ins and outs of how this kit works and the exact specifics of how to use it, when to use it, all those things, I would recommend going to the Miltenyi website where you can view that protocol there. I just wanted to say that we recommend it when you have a messy sample like this. So with our live demo complete, our nuclei isolated, purified, all of those considerations, we're happy with our sample. We're happy that it is a high quality sample based on all the analysis we've been able to do on the Celldrop, and we'll move to our downstream applications. So it seems like today the popular choice is RNA sequencing, but ATAC sequencing and flow cytometry are also other options here, and they all require a sample with plenty of nuclei, very little debris, very little intact cells. And you want your nuclei to be at a good concentration before you load them onto that chip as well. So you'll know the exact concentration of nuclei that you have, as well as the level of debris and intact cells in that sample. So you can be confident that when you go load them into these downstream applications, that it's a good sample. It's a quality sample that will hopefully give you good results in those applications. So in summary, I'd like to bring our attention back to what the Celldrop offers here. These nuclei extractions are lengthy, sensitive, and they're expensive procedures. There's a lot of equipment that we've talked about today. There's lots of kits and filters, and there's a lot that goes into these. And that's just to get to the nuclei isolation. That's not even to process those nuclei with RNA-seq or ATAC-seq. Some of those runs are upwards of $5,000 or $10,000. So you want high quality samples before you go off and run one of those experiments. Sample QC, sample quality control, is a critical component to ensure that you're not wasting money on a sample that wasn't extracted well or had some other issues to it that can be easily visualized by simply counting. So with image-based counting on the Celldrop, we are able to reproducibly QC all your nuclei samples in a way that is easy to interpret and powerful, that allows you to make quick decisions before you go on with your experiment. I'd like to also draw your attention to some additional resources. It's always impossible to cover all the information about something like this. So please visit Genovix.com. We have a series of ebooks. We have other webinars that cover other counting methods and counting protocols on the Celldrop. We have plenty of technical notes about different applications, including different nuclei isolation applications. And we have plenty more, like you saw our recommended protocol screen. We have so much to talk about in videos, so much to talk about that I can't cover it all even in one slide. So I would recommend visiting the website for any other information you would like about the Celldrop. And in fact, that QR code that you see on my screen right now can take you to all this information. So I'd recommend either screenshotting this slide or taking a picture with your phone so that you can go back and access this wealth of resources later. Most importantly, I'd like to encourage you to schedule a virtual demo or a free trial with myself or someone else on the application science team so that you can see how the Celldrop might improve your nuclei extraction workflow. So we can do something similar where myself or another member of the team hops on Zoom with you in a personal session, and we can work one-on-one with directly addressing your sample and your specific questions that apply to your workflow. Or more importantly, the best way to know if something works for you is to actually try it yourself. So we do a free trial program in the US where you can sign up for the free trial. We will mail you the device. We cover the shipping to you and back from your site. And during that free trial, you can work with myself. You can work with another member of the application science team to ask any questions, tune, optimize the equipment for your specific workflow and your specific samples. And then once you've been satisfied with your ability to count and quality control your sample on the Celldrop, you simply pack it up and mail it back to us when you're done. So that's also available through that screenshot or through that QR code on the screen here.